everyone. I'm going to get started. Thank you all for joining us for the August 2023 monthly seminar on physical genomics. My name is James Peterson, and I am the Senior Director of the Center for Physical Genomics and Engineering here at Northwestern University. CPGE is a transdisciplinary research center that is focused on studying the structure and function of the genome through the convergence of super-resolution imaging and nanosensing technology, modeling, computational genomics, biology, and artificial intelligence with a mission to develop new strategies for the treatment of disease and the reversible engineering of living systems, and to train the next generation of multidisciplinary scientists. You can learn more about our research and training programs at physicalgenomics.northwestern.edu or follow us on YouTube, Twitter, or Instagram. We are very excited to welcome as our speaker today, Dr. Nick Gilbert. Dr. Gilbert is a group leader at the MRC Human Genetics Unit and is Director of Graduate Research for the Institute of Genetics and Molecular Medicine at the University of Edinburgh. He started his career as a PhD student in the biochemistry department at Edinburgh University before doing a postdoc with Wendy Bickmore. He started his lab in the Edinburgh Cancer Research Center in 2006 with a fellowship from the Wellcome Trust and moved to the Human Genetics Unit in 2012 after obtaining an MRC Senior Fellowship. Dr. Gilbert, thank you so much for sharing your work with us today. For our audience, there will be time for questions at the conclusion of the talk, and you can type them into Zoom chat at any time, and we will address them during the Q&A after the talk. At this point, Dr. Gilbert, if you'd like to share your screen, you can. Yeah.
Okay, fantastic. Well, hi everyone. Thank you very much for inviting me to talk with you today. I was interested to hear Jay's introduction, and he mentioned super resolution microscopy. Now, I think I've got one picture of super resolution microscopy in my talk, but actually that's an area that we're doing lots and lots of work on, and I have to say, super exciting. But actually today, what I'm going to be telling you about are some of our sort of polymer simulation models to try and understand chromatin folding in mammalian cells. Now, what I'm going to do is I'm just going to try and share my screen, which hopefully will work. There should be something there. Right. So, yes, hopefully you can see that.
Yeah.
Now, let's see if I can get this menu bar out the way. Right, okay. So, what I'm going to do today is I'm going to tell you about some work we've been doing in collaboration with another lab here in Edinburgh, Davide Marinduzzo's lab. And so about 10 years ago, Davide and I started thinking about what would be the future of chromatin research. And one of the things that we thought is that actually the kind of questions we're wanting to answer are becoming more and more specific. They're much smaller questions, much more precise questions. We thought that maybe we're going to start getting to a point where actually experiments alone will be very, very challenging to answer some of the questions we're interested in. So Davide and I started thinking about what we could do to try and better understand chromatin organization. And Davide's lab is actually a polymer physics lab. And I have to be completely honest, it's taken us about 10 years of working together to really understand, me to understand the language of physics and him to become more of an expert in biology. But I think we're at a point now where we're actually able to combine our expertise and start thinking about developing polymer models that can help us to actually explain chromatin folding in mammalian cells. And what I'm going to do today is I'm going to tell you about some of our sort of latest models and how we hope we can use those models to actually understand something about chromatin organization and gene regulation in mammalian cells. So I have to be completely honest, I'm a molecular biologist. I understand some of the physics of what I'm going to tell you, but I don't understand all of it. So if you ask me questions that are too difficult, I might have to refer you back to Davide, but hopefully I can give you a flavour of some of the work that we've been doing. So let's just start off. So the level of chromatin organisation that we're particularly interested in is I suppose, the level of chromatin that's sort of around the scale of a gene. So 10 kb up to about 500 kb. And the reason why I'm particularly interested in looking at this level of chromatin organization is this is the level of chromatin organization that's absolutely crucial for gene regulations. In human cells, most genes are about 30 to 40 kb in size, and that's the sort of the coding region, and then that's then flanked by these sort of regulatory domains. And I think for us to actually be able to understand gene regulation, we need to have an sort of intimate understanding of all the components and the folding of individual genes. Now, we of course know a lot about the DNA. We know a lot about how the DNA is wrapped around individual nucleosomes, but we know far less about these higher levels of organization of the nucleosomes. And actually, one of the reasons why we know so little about it is because chromatin is a huge macromolecular complex, which is very difficult to study. And I think one of the challenges as well is that the organization of chromatin tends to be different in individual cells. So when we do a study on a population of cells, what we're actually doing is we're looking at the average organization between all of those cells. And I think to understand gene regulation, what we need to be able to do is relate the 3D structure of a specific locus and the transcriptional output of that specific locus. So I suppose our big questions are really to ask, what is the relationship between 3D gene structure and transcription? We know that individual genes are organized into coding units with promoters and enhancers, but actually, what is the three-dimensional organization of these genes? And, I suppose, one of the things I would love to be able to do before I retire at some point is to actually be able to come up with the 3D model of a specific gene. So you can actually see the shape of the promoter and you can see how the promoter changes when you activate gene transcription. Now, I used to think I had lots of time to do this work, but as every year goes by, I think, we need to crack on and do it a bit faster before I do retire. But I think that would be the really interesting thing, to be able to see the individual gene structures and then how they vary from one cell to another. And I think what we might find is that some genes have a propensity to adopt certain kind of structures, for example, ones that are more folded, whilst other genes will have more extended configurations. And they may well have evolved to be like this so that you can regulate genes in different ways. So what are the key questions? Well, what is the 3D structure of each gene? And how does this structure relate to transcription mechanistically? Now, many of you are probably doing experiments where you're starting to do sort of, single cell sequencing and really trying to understand the heterogeneity of processes like transcription. But it's still extremely challenging for us to actually be able to relate transcription in a single cell with the 3D chromatin structure in that cell. So what we wanted to do is to try and come up with a polymer model of chromatin that was able to enable us to see or to predict what the 3D structures of genes are, and for us to actually be able to predict transcription from those individual 3D structures. And that's what I'm going to try and tell you about today. So there are different kinds of models that people can use for chromatin, and the kind of model that we use is what we call a coarse-grained model. And so in a coarse-grained model, what we do is we remove quite a lot of the sort of information that's less important to our model. And this sounds like it's a way of sort of simplifying the chromatin, but actually this is really grounded in theories from polymer physics. And what we know is that the macroscopic behavior of polymers, for example, like chromatin, doesn't really depend on the specific details of the individual components, such as the nucleotides. So what we're able to do is take something that's very complex, like the individual nucleosomes, how they're connected together along the chromosome, and actually simplify this and coarse-grain our model. So the kind of coarse-grain model of chromatin that we use is what you'd call a bead spring polymer. So essentially we model chromatin as beads, and these individual beads are then connected together by little springs, and we put in the biophysical parameters you would expect for chromatin, so for example the correct persistence length, but we don't model individual nucleosomes. Instead we use about a resolution of about 1 bead per 1,000 base pairs. Now sometimes we have done models where our beads are slightly higher resolution, maybe 1 bead corresponds to 400 base pairs, but actually we find it doesn't make a huge difference in terms of the results it gives us, and it takes much longer for us to be able to do the simulations. So when I'm going to talk about beads in my presentation here, then the beads essentially are equivalent to round about 5 individual nucleosomes. So there are different kinds of simulations that people use in the chromatin field. So one of the kinds of simulations that you often come across are what people call inverse modelling simulations or top-down simulations. Now these have been very helpful in enabling us to understand chromatin organisation, and what people often do is they take complex 3D structure data, for example high-C maps or 3C contact maps, and from that they're able to fit the 3D structure of a gene to this experimental data. And this is a very good way of actually being able to visualize, for example, the 3D folding of chromosomes. But we decided to use a slightly different approach, and what we use is mechanistic modelling. And the reason why we use mechanistic modelling is we don't want to put any experimental data in our model. We want our model to be completely predictive. And by doing this, we can actually use our models to both simulate the 3D structure of genes, but we can also use it to actually help us generate hypotheses for actually understanding chromatin organisation. So for example, the kind of modelling that we do, that we call mechanistic modelling or bottom-up modelling, we start with our coarse-grained model of chromatin, and we apply all these biophysical principles to the interactions that happen on the chromatin fibre. And from this, we can then predict structures, and then to test our model, we then make simulated, for example, interaction matrices that we can then compare to published high-C data to see how well our models work. And we also combine it with fish, where we can use individual fluorescent probes and we can then simulate what the fish data would look like, and then we can do the experiment and see if the two compare to each other. So in all the models I'm going to show you today, we don't put 3D structure data into our models. We've just got fundamental principles of chromatin folding, but from those simulations we're actually able to infer the 3D structure, and by doing this we're actually able to try and start understanding what the individual molecular mechanisms are that are important for chromatin folding. And actually one of the exciting things about our modelling is sometimes what we find is that our simulations don't really fit experimental data, and then that tells us we need to modify our models to have different properties or behave in different ways so they do properly recapitulate the experimental data. So the way our sort of model works is we start off with our bead chain polymer and the individual interactions that happen on the polymer. We then do simulations and we're able to generate the 3D structures of the genes. We test these experimentally, and from this we then formulate hypotheses to reconcile the discrepancies, and we're then able to generate new models. So we've got this constant cyclical process. And one of the other things we're really trying to do at the moment is actually use our models of chromatin folding to make predictions that we can go and experimentally test. And I think this is particularly important because, as many of you know, actually doing the laboratory experiments is extremely time consuming. And so you can imagine engineering a gene locus with CRISPR to move around some regulatory elements. It's probably going to take you 6 months to do that. And if you spend 6 months and the results aren't really what you're interested in, then that's very, very frustrating. So one of the things we like to do with our models is we like to be able to do in silico tests of our experiments before we actually do the experiments to see if they're likely to give us the kind of results we're actually looking for from our studies. Now, we're only just starting to do that, so unfortunately I don't have any data to show you, but one of the things we're really excited about is whether we can actually put, for example, CTCF sites at different positions in a gene locus and then alter the 3D structure of a locus and then affect the transcriptional properties of that locus. And that's something that we're trying to do at the moment. So the kind of model that we've been using to model chromatin we call HIP-HOP, and it stands for Highly Predictive Heteromorphic Polymer Model. And so we've spent about 10 years developing this model. First of all, from a sort of fairly simple bead string polymer, and then we've added extra components into this model. And what I'll do is I'll talk you through these individual components separately. But essentially what we have is this bead string polymer where each bead is about 1 kb in size. And we start off by painting these beads with the underlying epigenetic marks. And so this is the only experimental data that goes into our model, and this is the position of epigenetic marks. Now, the marks that we put into our model are the ATAC binding sites, so those are reflective of regulatory elements. We have H3K27 acetyl marks, and those are reflective of active chromatin. We've got H3K9 trimethyl, which is a mark that's a reflective of repressive chromatin, and then H3K27 trimethyl, which is then a mark of polycomb chromatin. So the way our model works is it's predicated on chromatin binding proteins being multivalent that form 3D interactions within the fibre. And this happens through a process called bridging-induced attraction, and I'll tell you a little bit more about that in a minute. But this is one of the key components of our model, this process of chromatin binding proteins driving the 3D structure. In addition, in our model, we have loop extrusion. So we simulate loop extrusion and cohesin being loaded at CTCF sites and then extruding loops out on the chromatin. And then the last bit of our model that we found was absolutely crucial is we need to model 2 different levels of chromatin organization. So this is why we call it a heteromorphic polymer, because essentially what we found is that gene active regions that are marked by H3K27 acetyl are much more flexible than inactive chromatin. And so what we have is in active regions of chromatin, we just have single springs between the beads. But in inactive regions of chromatin, we actually have 2 springs between the beads. And this is a way of stiffening up the chromatin fibre in regions that aren't transcriptionally active. It also has the effect of meaning that transcriptionally active chromatin has essentially got a few more disruptions and is more flexible and is more able to adopt the 3D configurations. So one of the important principles of our model is this process called bridging-induced attraction. And the way it works is we essentially model our bead spring polymer, but our bead spring polymer has binding sites for multivalent binding proteins. Now, the chromatin doesn't have an affinity for itself, but it does have an affinity for these multivalent binding proteins. So we have certain class of repressive proteins that bind to H3K9 trimethyl marks, and we have certain kinds of active proteins that bind to H3K27 acetyl marks and the ATAC binding sites. And what happens is these individual proteins, diffuse around the chromatin, and then they interact with sites that have been painted with our epigenetic marks. And what we find is that you actually form these multivalent interactions which start forming these 3D structures. And this is very important because it's this process that actually drives the 3D structure of the chromatin. Now, the way it works is if you've got a binding site, a protein that's floating around, it will bind to the sites that its normal targets are. And it will form these little connections in the fibre. And this then has the effect of increasing the concentration of chromatin in these regions, which then induces more factors to actually be able to bind. And so what we've realized is that actually within chromatin you form what we call microphase separated structures. So this is not like the big phase separation you hear people talking about with big transcription factor complexes. These are microphase separated structures where you might have 3, 4 or 5 of these regulatory elements coming together. And I'll show you later on some of our simulations, and you can actually see how these regulatory elements actually cluster together and help drive the conformations of our individual polymers. So very importantly, the transcription factor binding leads to this bridging induced attraction that drives the 3D structure of our locus. And as I mentioned, we have different classes of transcription factor in our model. We've got active marks that bind to the ATAC sites and H3K27 active chromatin. We've got polycomb marks and we've got repressive marks. And I suppose if you wanted to think of specific proteins that might behave in this way, so the repressive proteins might well be proteins like HP1. The polycomb proteins might be the ones that bind to H3K27 trimethyl, and it's proteins like CBP P300, for example, that will bind to these active marks. Now, I draw these transcription factors at single beads, but they're multivalent, and we actually don't think they're probably single proteins, but there'll be small clusters of transcription factors. So it'll be 3 or 4 transcription factors coming together in a little complex. And then they've got this multivalency and able to bring together 2 bits of chromatin. So you can see here where we've got 2 different domains formed from the active marks that are binding to active regions, and another domain where you've got the repressive proteins binding to inactive regions of chromatin. And so this would be analogous to A compartment and B compartment if you're making an analogy to Hi-C. And so these then form these little microphase separated structures. But we're not talking about big microphase separated structures. There's probably just a small handful of individual components, and they are very, very dynamic. We had a paper we published just last week where we actually predicted how quickly the proteins pass in and out of these structures, and they last for fractions of seconds. So constantly you've got proteins coming in and proteins coming out. So these are, when you look at a snapshot, they look static, but actually if you run dynamic simulations, then they're very, very active structures that are inside the cell. So the other component we have in our model is loop extrusion, and the loop extrusion is important because this drives a higher level of organization. So you can imagine that the bridging induced attraction is important for driving interactions between enhancers and proteins, whereas loop extrusion is important for driving a slightly higher scale of chromatin organization. And the reason for that is obviously there are slightly fewer CTCF sites, they're often a greater distance apart, but they're driving these higher levels of organization. Now, when we do our simulations, we can actually predict the high-C structures, and we can see the kind of features you would expect to see in experimental high-C maps. We can see the same features from our simulations. So, for example, you can see little hotspots in our simulations where the CTCF binding sites are actually positioned on the chromatin fibre. So what we wanted to do is make a baseline map of the 3D structure of the whole of the human genome. And so to do this, we decided to pick a cell line called GM12878. This is a human lymphoblastoid cell line. And the reason why we picked this cell line is it's a tier 1 ENCODE cell line. So there is lots and lots of experimental data and high quality epigenetic data available for this cell line. And so remember, the marks that we wanted were ATAC, H3K27 acetyl, we wanted H3K27 trimethyl and H3K9 acetyl, and we also wanted to see the underlying CTCF binding sites so we could specify where the loop extrusion was going to happen. Now, in our simulations, we're not able to simulate the whole genome all at once, and so what we did is we essentially took the genome and we fragmented the genome into different blocks. So you can see chromosome 1 here, we couldn't simulate it all in one go, so we've got 1, 2, 3, 4 segments that we've simulated chromosome 1 in. For some of the smaller chromosomes, like the ones after chromosome 14, we were able to simulate them in one go, but the bigger chromosomes we couldn't. And when we do the simulations, we don't just simulate each genomic segment once, but we simulate each chromosomal segment 600 times, and this gives us a panoply of the different structures that the chromosome is able to adopt. So this is an example for our data for chromosome 20. So you can see this is the chromosome 20 and we've simulated it 600 times. So we've got 600 pictures of what chromosome 20 might look like it's actually able to adopt inside the cells. And then once we've done the simulations, we can then abstract the individual genes from the chromosome. So we can look at whole chromosomal segments in one go, but for this study, we were particularly interested in then extracting out information from for individual genes. So this is an example of a gene called CD40, and then we're able to extract all 600 structures that this individual gene is able to adopt. Now, I have to be completely honest, computationally, this was a huge amount of work. I pissed off so many of my colleagues because we basically had to monopolize our supercomputing cluster for over a year for doing the calculation. This was 10 million core hours of processing, and so very, very computationally intensive to do this. So now that we have a baseline, we possibly won't re-simulate entire genomes, and instead we'll probably just look at individual chromosomes, because it would take us a whole year if we were to try and do another cell line, and that's obviously a huge investment in time and resources. So this is just a zoom in of human chromosome 20, and what we can see here is that the chromatin is coloured in grey, and then different features of the chromatin are coloured in different colours. So for example, you'll be able to see regions that are yellow, so these are actually coding regions on chromosome 20, and in red are the ATAC binding sites. Pink are polycomb regions, and blue is H3K9 heterochromatin. And just by looking at this, even at this very, very crude scale, you can see that the heterochromatin is tending to coalesce with itself and forming an individual structure. The individual genes are much more spread out through the organisation of the whole chromosome. And then if we have a look at the individual gene, so this is CD40, we can then look at the individual beads. So what we've done here is we've smoothed it a little bit just to make it a bit easier to look at, but you can essentially imagine these are our individual beads in the chromatin fiber. And you can see the sort of, the coding regions, and you can see the individual ATAC binding sites. But I'll show you on our 3D gene software in a minute for a couple of other examples so you have a better feel for it. So once we've done the simulations, what we wanted to do is then validate our simulations. And the way we do the validation is we use FISH. So we take fluorescent probes and we then predict where these fluorescent probes will be in 3D space and the measurement between these probes. And then we do experiments and we compare our experimental data with our simulation data. And so this is just some examples from the X chromosome and chromosome 11, where we've picked probes that are either about 2 megabases apart or 1.5 megabases apart. And we look at the simulated FISH data, and that gives you these distributions in blue. And then we've done the experiment, and you can see there's a lot of similarity between these two structures. Now, we're in the process at the moment of actually doing about 20 more gene loci to try and increase the numbers. So far we've done about 10 gene loci. It's unfortunately quite time consuming doing the FISH, but we're trying to increase the numbers. But what enables us really to look at our genome-wide maps and compare them to experimental data is Hi-C. So GM12878 cells, we're very fortunate, people have generated very high resolution Hi-C data sets, and we're able to look at the simulated Hi-C to the experimental Hi-C. And this is shown here. So this is for a 10 megabase region on chromosome 12, for example, but we could have picked any region on the genome. And you can see that actually our simulated data is very, very similar to the experimental data. And pretty much everywhere we look at across the genome, we have very, very good relationship between the two. Now, we wanted to quantify this, and we came up with an algorithm that enabled us to calculate something we call domain directionality, and we were then able to plot the interactions between different regions, and we were able to compare what our experimental results were to the simulated results. And so this is some of the data down here. The simulated data is in red and the experimental data is in blue, and you can see these lines are very, very coincident with each other. And the paper we have on bioRxiv, we've shown this data for many other chromosomal segments. So what I want to do now is I just want to be able to show you our 3D gene database we've generated. So essentially what we've done is we have taken the 3D structure of all active genes in GM12878 cells, and we've now put them into a database so people can actually look at the 3D structure of the genes that they're interested in. So what I'm going to do is I'm just going to switch to a browser so I can show you in real time. So hopefully you can see that on your screen. So this is our 3D gene database. This is the address up here, 3dgene.igc.ed.ac.uk. A little bit of an introduction about it. We've got an FAQ that tells you what all the colours of the different beads mean. But the important thing is you can go in and you can actually put in the name of your favourite gene or the ensemble ID for your favourite gene. So let's have a look at a gene called PAC6. PAC6 is a really important gene that's important for eye development. It's considered a master regulator, but it is expressed in these lymphoblastoid cells that we're looking at here. And we search for it, gives the location, and we've already extracted that gene in our database, and we can actually then look at the 3D structure of this gene. And the software that we're using at the moment for visualizing it is actually software that's used for looking at the 3D structure of proteins, but you can then rotate and see the 3D structure of your gene. Now what we've also done is we've uploaded 3 different topologies. So that's topology number 1, the most common topology you find for that gene. This is number 2. So you can see it's got a different configuration. This is the second most common topology you find for this gene. And then we can look at, for example, topology number 3. And you can see in this topology here, you've actually got a more extended structure where it's not quite so globular, but it's more open. Now, we also have a downloads page. So if you're interested in looking at any of the 3D data yourself, you can download our raw data files that detail all the interaction coordinates, and you can essentially just go in and put in the gene regions that you're interested in and download all of our data for all 600 different simulations in SIF format to be able to look at your regions of interest. And what we also show is the contact matrix that we've simulated for these individual genes. So this is our simulated contact matrix for the PAX6 locus. Now, of course, the simulation matrix, it's actually an average between all of our 600 different structures. So, please give it a go. We're still trying to work on coming up with different ways of visualizing these different 3D structures. But this is essentially the data that comes from our simulation model using the hip-hop framework. So I'm just going to go back to my talk. So once we'd simulated the 3D structure of all of the genes in the genome, we wanted to start data mining it. Now, I have to be completely honest, we're still in the early days of data mining all of this information. And I love the fact that the lecture series here is called Physical Genomics, because this is completely what we're looking at. We've got these physical structures of these genes, and it's a huge reservoir of information. But we had to start somewhere for actually asking these questions. So the first thing we wanted to ask about is looking at gene interaction partners, and we were able to go into all of our data, and we were able to ask how frequently the promoters actually interact with the ATAC binding sites in a locus. And then we were able to score this interaction frequency. And we came up with this idea of influential nodes. And so influential nodes are sites within a gene which preferentially interact with a promoter. And we found actually some genes have a small number of influential nodes which drive the 3D structure, whilst other genes have far fewer. And so this is a few examples of what we see. So this is just taking 3 different genes from our data set. So this gene here, ZBTB5, has very, very few influential nodes. So what that means is there are lots of parts of the gene which are actually important for driving the 3D structure. Whereas this gene here, TERF1, what we find is that it has a very, very strong ATAC site that drives the 3D structure of the locus. Now, when we were doing this analysis, we actually came up with a new word for a 3D structural unit, because we're not really talking about genes here, because we're actually talking about the whole regulatory domain for a locus. So this region here, rather than referring to it as a gene locus, we now refer to it as something called a topos, which is a Greek word meaning a sort of 3D topology. And then the plural of that is topoi. So this really gives a new concept. So instead of thinking about a gene as a structural unit of heredity, what we think of is a topos being a fundamental unit of 3D structure for an individual gene locus. So this is one feature we've realized, is that influential nodes are very important for driving the 3D structure. And what we find is that actually genes with influential nodes tend to be linked to genes that are important for developmental processes. So the cell line we're looking at here are lymphoblastoid cells, and it's an immune cell type. And actually the genes with influential nodes where we think they're driving the 3D structure are actually linked to the Go terms. And what we find is these are overrepresented in genes with the influential nodes. So we don't fully understand this relationship, but we think influential nodes are linked to the developmental importance of genes in a certain cell type. So one advantage of doing our simulation 600 times is it enables us to look at what we call structural diversity. So we're able to look at these 600 structures and look at the heterogeneity that individual genes are able to adopt. Now, what we mean by this is the number of different unique topologies we actually see within a locus. We had to come up with a definition of a topology, and you can see in the little cartoons here, in this example here, the promoter is interacting with ATAC site A, whereas in this example here, the promoter is interacting with ATAC sites B and C. And we're able to then score the number of topologies for these individual genes. Now, what we do see is there's a clear relationship between the number of partners or ATAC sites and the number of topologies. And this isn't surprising. Genes that have got more ATAC sites are going to be able to form more of these individual topologies. But if you then look for genes that have the same number of partners, so let's say 20 partners, you've then got this huge range of different topologies they can adopt. So let's just show you a couple of examples. So if we look at two different genes, SMARCA5 and GINS4, they both have the same number of ATAC binding sites, but actually GINS4 is able to have far more of these different topologies than SMARCA5. And so let's have a look at that. So for SMARCA5, it's got 8 interaction partners and has about 56 topologies, but of these topologies, there's one that's a very, very dominant topology, whereas GINS4 has 8 partners, it's got a few more topologies, 89 topologies, but you can see that actually there are very, very few dominant structures. So we would say SMARCA5 is a low diversity gene because it's predominantly structure 1, whereas GINS4 is a high diversity gene. And we think this is important because what we think it means is that if you look at a population of cells, SMARCA5 is going to have a tendency to always adopt the same 3D structure, whereas GINS4 is able to adopt many, many different structures. And I'll come to this in a moment, but we think this is then linked to transcriptional heterogeneity, because you can imagine in a scenario here, you're only going to adopt one structure and then you will essentially just form a locus that's more likely to be transcribed in the same way every time because it's got the same structure, whereas a locus that's able to adopt lots of different structures might be transcribed and regulated slightly differently. And so we can see this very strong relationship between influential partners and diversity. So these influential nodes are linked to the diversity, and the more influential partners you have, then the more diversity you've got essentially in this relationship. So what we wanted to then do, and I'm just going to take a very, very side step in just telling you about something else we're now looking at in our data, and this is a very, very new thing, is we wanted to try and understand how genes cluster within our simulations. And I haven't really got time to go into the detail of this, but in another project, what we've been able to show is that a protein called HNRMPU is an abundant nuclear protein. It binds to RNA, and what we've been able to show is that it essentially organizes itself around individual gene units, and it's important for decompacting those individual gene units. And if we do super resolution microscopy for H&R and PU, what we can see are all these little clusters inside cells. And we hypothesize that these little clusters actually correspond to maybe 1, 2, or 3 genes coming together and forming these little micro-compartments. And so one of the things we're doing at the moment is we're doing experiments to look at these individual clusters, but we've also started going into our simulations, and we can see these little clusters being formed in the simulations. And so this is our little model of a cluster where we've got maybe a small number of genes coming together. These transcriptionally active genes make nascent RNA that interact with H&R and PU and form this little micro mesh around, forming this little microenvironment around active genes. And the size of these little clusters from experiments is about 100 nanometers in size, and then by our simulations we can look at our simulation data, and these clusters are also about 100 nanometers in size. So this is just diameter along the x-axis here, and this is frequency on the y-axis. So we think we can then go into our simulations and it can actually tell us about how genes are interacting within the genes are interacting together within the nucleus. And I haven't unfortunately got the graph to show you here, but if we then look at the number of genes in these individual clusters from our simulations, we can see generally they are just a single gene, but then some of them do have 2 genes, 3 genes, 4 genes, right up to about 8 or 10 genes that are coming together in these little 3D structures. So this is way, way smaller than a TAD, but it's a sort of sub-organization, if you like, of genes coming together inside the nucleus. Now, the last thing I want to touch upon in my talk is transcription. And when the physics guys told me that they thought they could predict transcription from our simulations, I really thought that was completely impossible. So I was so skeptical when we started doing this analysis. But amazingly it does work, and I'm going to show you the data to demonstrate that. So what we decided to do, and the way we decided to simulate transcription, is we put into our model essentially an active transcription factor, and we said when the active transcription factor comes in contact with a gene, it then fires. And in our simulation, we then score the number of times that gene fires. Now, the way we do our simulations is our simulations run through about 10 million time steps. So every time step, we recalculate the 3D structure of the gene. But for the last 1 million time steps, we predicted the transcription for each individual gene. And so what this means is for every gene in our simulation, because we've done the simulation 600 times, we have a transcription value of, we have 600 transcription values for each of our individual genes. So this is just some data for 2 genes we've picked, KCNB1 and CNBP. KCNB1 is a more lowly transcribed gene, and we can see that it doesn't have as many firing events as CNBP, which is a more highly transcribed gene. And so what we can do is we can then simulate the mean transcription from our 600 individual simulations, and we can compare it to experimental data. So the experimental data I'm showing you is published data, and it's gross-seq data. So gross-seq data is nascent transcription data. So on the x-axis, we have experimental transcription data, and on the y-axis, we have our simulated transcription. And you can see the correlation between the two, and we've got an R value of 0.57. And so this is us simulating the level of transcription purely from the 3D structure we've simulated. We've put no transcription information into our simulation. We've got no 3D experimental data in our simulation. all we've done is painted the chromatin with the epigenetic marks, and we're able to predict transcription as well as we are. And as I mentioned, this is published data. What we've just done is we've done a whole series of single cell sequencing experiments ourselves, and we're now correlating our simulation data to our own single cell sequencing data, which we hope will be even better relationship to what we've got here. But the great thing about actually being able to look at the individual simulation data is we can then actually go into our simulations and say, well, why is one gene more highly transcribed than another gene? And so the first thing we notice in our simulation is there's a relationship between the number of bound transcription factors and mean transcription. So you'll remember right at the beginning I talked about microphase separation, and what we find is that highly transcribed genes, these active factors, have a tendency to come together and form these microphase separated clusters, which increases the possibility of gene firing. So if we look at our two genes here, KCNB1, which is lowly transcribed, and CNBP, which is highly transcribed, for CNBP, we're not really forming a very big microphase separated cluster around the promoter, whereas KCNB1 we are, and sorry, CNBP we are. And so this is actually us dynamically forming these microphase separated clusters, and this is what's really driving the high levels of transcription in our simulation. But what we're also able to do from our simulation is, because we've done the simulation 600 times, we're then able to go into our data and we're able to look at how noisy a gene is. Now, what we mean by noisy is how heterogeneous the transcription is. So if you imagine you've got a low noise gene, that means each individual locus is essentially transcribing the gene at a similar amount. Whereas a high noise gene, you've got this very broad spread of transcription. Now, actually calculating this from experimental data is virtually impossible. We are trying to do it using 10x Genomics, and we've just started using another single cell sequencing kit from Past Biosciences, but it's extremely difficult. But we can do this with our experimental data. So this is just data for two genes, SMARCA5 and GINS4, and you can see that GINS4 is a much noisier gene. When we look at our 600 individual simulations, there's much more heterogeneity between these individual simulations, whereas SMARCA5 is a much narrower relationship. Now, the really amazing thing is that this then correlates to structural diversity. So genes that have a low level of structural diversity have a lower level of transcriptional noise, whereas genes that have a high level of structural diversity have a high level of transcriptional noise. So this goes back to what I was saying to you earlier, that if you've got influential nodes which drive specific 3D structures, then that's likely to be a less noisy gene than a gene that's got lots of regulatory elements that's able to adopt lots of different 3D structures. And we think this might be important for evolution, because what you find in evolution is old genes tend to be less noisy than young genes. And so what we think is, during evolution, you tend to find these influential nodes that then determine the 3D structure of a locus. And that's something we'd love to be able to test experimentally by essentially making a synthetic locus and seeing if we can vary the level of transcriptional noise. So what I've told you today, and I've whizzed through a lot, and I apologize, but you know, there's just a couple of very specific messages that I'd like you to be able to go away with. So the first thing is that transcription factors phase separate to form these microphase separated clusters, and this drives mean transcriptional activity. So highly transcribed genes, you've got these regulatory elements coming together in the proximity of a promoter, and that's able to induce these microphase separated clusters and increase the firing rate of an individual gene. And so we can see that in this little model here, where a low activity gene, you are forming very few of these microphase separated clusters, but a high activity gene, you're forming this microphase separated cluster in the vicinity of a promoter. And the other thing I told you, and I'm sorry I don't seem to have a picture for this, is we can then see this relationship between transcriptional noise and structural diversity. And the more different 3D conformations a gene is able to adopt, the more different levels of transcription for that individual gene. So that just leaves me with the acknowledgements. So all of the simulation work was done by Michael, who was a PhD student in Davide's lab, and he worked very closely with another group leader, someone called Chris Brackley, and Michael is now doing a postdoc with Davide and me, and he's an absolute whiz at doing the simulations, but importantly is for us to do the experimental validations, and two of the people in my group, Catherine and Ruski, have done a lot of those. The problem is that the computer guys can do the experiments so much faster than the wet lab folk. So I'm constantly feel embarrassed because they come up with these new hypotheses and then they say to us, well, you know, can you do an experiment to test it? And I say, yep, of course we can do an experiment, but it'll probably take us about 5 years to actually do it. So it's unfortunately it's the biologists that are really holding the physicists back here. But I'm hoping they can tell us the right experiments to do. So thank you very much for listening, and I'll be delighted to answer any questions that you've got.
Fabulous. Thank you so much, Dr. Gilbert. If there are any questions, please feel free to just unmute or put them in the... So Gary Carpin entered a message in the chat. Wonderful talk, Nick. Excited to see more integration of polymer simulation with the entire genomes and experimental data regarding bridging biolimitation simulations to microphases. Given that HP1, PC, BRDs form large condensates, does inclusion of macrophases impact the results, e.g. improve correlations with experimental data in those regions?
So yeah, no, that's a really good question. And so we have done other simulations, particularly to look at larger condensates. But using the parameters that we're using at the moment for these simulations here, we don't see these larger condensates forming. And I'm not quite sure why we're not really inducing them in our current simulation models. It may well be that because we're not really doing the simulations in a nucleus, then the chromosomes are much more able to freely move around, and we don't have the sort of subcompartments in a nucleus, like for example the nucleolus or the nuclear membrane, which might be an additional factor that's able to start driving these larger condensates being formed. But I think that'd be a really interesting for us to think about and actually bring into our simulations when we're looking at chromosome scale.
Fabulous. Xiaomin Bao has a message. Great talk. I have 2 questions. Was wondering if you are planning to include chromatin associated RNA in the simulations?
So yes, absolutely correct. So that is the thing we're working on at the moment, and we have got a paper that we are, well, hopefully I will get it on bioRxiv in the next month. And what we have essentially started to do is introduce RNA into our simulations, And actually, maybe if I can just share my screen again, where's the share button? I can show you the way we're doing these simulations. You can see this is me actually drawing my figures for the paper. Where is it? So what we're essentially doing, oh, maybe it's not going to work. So I don't know whether you can see this. Maybe it's a bit too small, but essentially we're trying to develop new simulations where we're trying to include an RNA component. And we think forming these sort of, protein RNA structures actually has an ability to decompact chromatin. And the reason why we think this is because chromatin has a natural tendency to collapse in on itself. But if you form a protein RNA gel around a gene, it then competes against that desire for chromatin to collapse and therefore has an effect in opening the chromatin. So at the moment, those simulations are done with a slightly different model. We haven't introduced them into the hip-hop model yet because that's more complicated. We're doing a slightly more basic model of chromatin folding to develop those simulations. But yes, that is the next evolution of hip-hop, is to bring in this RNA component. And so what we'll then have is our nascent RNA coming from the promoter and forming this cloud of RNA that's then forming these little gels which will help decompact the chromatin. So yes, that's what we're doing.
And so, Xiaomin, second question, with the prediction of transcription, what are the features of genes that show the lowest prediction power?
So what we find is, and I didn't have time to show you this today, but CTCF and loop extrusion does not correlate to transcription per se, but it does correlate to transcriptional noise. So genes that have quite a lot of CTCF sites and are doing a lot of active loop extrusion are able to adopt lots of different 3D structures, and that affects transcriptional noise, but it doesn't seem to affect the mean level of transcription. The thing that's most important for correlating to mean transcription, it seems from our experiments, are the number of ATAC binding sites, so the number of enhancer elements, or the strength of an enhancer element. So if you have, just one very, very dominant enhancer element, then that also drives the level. So it's an interesting interplay between the promoter and the individual enhancers. And these are things that, I'll be completely honest, we've only just touched the surface trying to mine the data out of our simulations, and we probably need to come up with better ways of thinking about doing the analysis.